studying pigeons, you can learn about jaw development, facial development in humans, because it's pretty well conserved. Okay. So this is kind of where we were ending last class. We were trying to describe genetic models for development and not thinking about the specifics. This is a fly picture, but what general properties can we learn? And the most general property is that the development in the adult is really laid down early in development, so this is an embryo, by gradients of gene expression. So development is all controlled by gene expression. Gene expression doesn't occur randomly. It occurs in gradients that lay down a body plan that will kind of set the stage for further development. And then so we had a clicker question based on this, right? So you have, and the point of this clicker question, or there were 2 points. The first point was that these gradients, it's usually not like presence of a gene and absence of a gene that define the structure. They're usually complementary gradients. So here in the anterior portion of the embryo, so in the front, you have this gene called bicoid. On the posterior, you have a gene called nanos. It's not the absence of bicoid that gives you a posterior, it's the presence of nanos. And there's a bunch of genes in between. So this is just kind of the 2 poles, a bunch of genes in between. So the clicker question asks you just, even though I have just said this, like reread it, look at the data, because you should be familiar with thinking about data like these. What do you see? And then I added another clicker question, if the expression changed, right? It wasn't a clicker question, it was just a question for you to think about. If now we see expression throughout the whole embryo, what type of mutation caused that? And so you should have discussed that in the discussion yesterday, right? We have certain types of mutations, deletions, insertions, SNPs. They can correct coding regions, regulatory regions. There's only so many types of mutations that can give you a pattern like this, from constrained expression to big, constitutive expression in all tissues. Or the flip side, there's only so many types of deletions that can go from expression in one specific area to no expression. And I said that's going to be on the exam. So that was all kind of a quick wrap-up of what we did in terms of development last time. And so now we're going to move on to a couple more cases of development. And they are Drosophila specific. You don't have to memorize the specific aspects of the development of Drosophila, but it's to think, what are you learning about genetics by thinking about development in these contexts? So in the embryo, so we're switching where we are in the Drosophila life cycle. So here, we were in, I cut out the slide with the life cycle, right? It's an egg, an embryo, then a larva, a couple larval stages, then a pupal stage, then adult. It's a kind of a classic insect stage. So these are all embryonic genes controlling polarity. So laying down where structures will occur, and now you have to start adding in structures. So this thing here is the larva. And what you can see is it looks like it has all these segments, right? If you think about a caterpillar is a larva, it has kind of segments. Right? People have learned a lot about development of segments and how different genes actually, what makes a segment? It's not that you have unique gene expression in each individual segment, it's that you can kind of split genes into 3 rules. So this is why they're called the rules on the bottom. So you have gap rule genes, pair rule genes, and segment polarity genes. So these are just examples. So the main take-home point is that even though we're going to end up with a really complex development, it's not that you need tissue-specific expression for every tissue. You can combine genes in interesting ways and look for overlapping patterns to generate certain tissues. So in these gap genes, they're really determining really large regions of the embryo. So kind of head versus thorax versus abdomen, right? So a larva does not, it doesn't really have a head. Those are what are going to occur in the adult. So head, thorax, abdominal segments of the adult. If you have a gene that's responsible for making these gaps, they're named by the mutant. So if you have a mutation in this gene called Krupel, you kind of lose a whole segment or a whole set of segments that correspond to a body plan. So that's why it's a gap. So the colors are really hard here with this Krupel mutant. So I'm just going to write it just so you can, in case it's hard to see. So this mutant is missing the dark blue and yellow. So it's like you created a gap. You kind of fused now the latter half of the abdominal segments with the head. You kind of chunked out a whole gap by having a mutation in this gene called Krupel. Krupel is a German word, I forget what it means, but those are examples of gap genes. They have really big detrimental effects on fitness, right? You can't have an adult fly that's missing a thorax. It's just not going to work. The next set are called these pair rule genes. And so these genes, they don't delete whole chunks of the embryo, they delete alternating segments. So that's what's trying to be shown here in this gene called even skip, right? You still have yellow, dark blue, and light blue, but now you're kind of missing alternative segments. So that's what's in that yellow box. Mutations of even skip cause the deletion of an even number of segments. And the last one is a segment polarity gene. So another example. So usually you get a very specific polarity, and now you're just flipping the polarity. So that's shown in like those black stripes. You're still having the thorax segments and the abdominal segments, but you just reverse the polarity. Is that important for you to memorize? No. What's important for you to think about are the patterns, right? The identity of thoracic segment number 3 is due to not a unique gene that says you are number 3. It's due to an overlapping expression of Krupel, of even skip, and gooseberry, right? So these are the mutant phenotypes, but from mutant phenotypes we can infer biotype function. So to get thorax segment number 3, you need the expression of all those genes. And those aren't going to be the same, right? 3 genes, all these permutations. This is not a complete picture, but I'm just, as an example, can give you so many different varieties, just 3 genes, right? Does that sort of make sense? Okay. So the last developmental structural genes we're going to think about in Drosophila are called homeotic genes. So what is a homeotic gene? So we're talking about these segments. Homeotic genes identify segments. So there's more to this definition, but we're going to explore that definition through a top hat question. So you can understand what a homeotic gene is by thinking about the consequences of a mutation in a homeotic gene. So this should have been covered in your reading. So mutations in homeotic genes often cause the deletion of segments, the absence of structure, too many segments, or structures to appear in the wrong place. So Krupel is not a homeotic gene, so don't think about Krupel, even skip, gooseberry. Those are not homeotic genes. We're kind of transitioning to the last class of developmental genes that we're going to think about. So let's start ruling some of these out. What do you guys want to rule out? And because? Okay. Yeah, so homeotic gene, those other genes we saw, they're missing segments. This is not about the number of segments, it's about the identity of the segment. So you can kind of rule out C for the same reason. We're not going to add or remove segments. We're saying what is the identity of a given segment. So what does that definition apply now? We have B or D. Does that definition imply that things will be missing or things will be in the wrong place?
In the wrong place.
Yeah, I think so. So the best answer for me is D. If you're going to identify a segment, and now you, by gene expression, and now you have a mutation that's going to change where a gene is expressed, you're not going to lack a segment, you're going to change the identity of the segment, right? It's again, this is not usually caused by the absence of gene expression lacking a structure, it's the presence of gene expression causing a structure. So this is one of the most famous examples that we put in textbooks. This is a mutation in a gene called antennapedia. So I don't know if you can tell, this is like the normal wild type fly head. And this one has a leg, right? That's a leg coming out of the fly head. You change the expression, you have a mutation in a tenapedia that changes the expression domain where this gene can be expressed. It's obviously usually expressed in thoracic segments, that's where the legs are going to be. Now it's expressed where the antenna would normally be, and you get a leg. So that's how they named the gene antenna. Put a leg where your antenna goes. Does that sort of make sense? So why is that important? Knowing what type of developmental gene you're dealing with, whether it's a gap gene, pair gene, homeotic gene, helps you make predictions. That's the whole important part, that you can go kind of back and forth. You can make mutations, look at data, and say, okay, I understand how this gene affects development. Or you can go the other way. I want to understand how this structure is determined, how development is determined, what genes lead to this structure. You can go back and forth, but you have to always go through the logical argument that certain types of genes should give you certain patterns of expression and formation of structures. So the really famous chunk of homeotic genes are called Hox genes. They come in two kind of clusters. Or Hox genes is what we call them in other animals. In Drosophila, you have the antennapedia complex and the bithorax complex. So what's really interesting about these is that they're tandem duplicates, right? We've seen that before. There's some nature, these genes are all close together. They're inherited as a unit. I don't think all of them are necessarily duplicates, but I think people have actually found out that they are kind of tandem duplicates. We talked about the role that duplication plays in the evolution of new genes. Well, it kind of makes sense, right? If you have a gene that's really important in segment identity, and it gets duplicated, and it takes on a new promoter, transcription factor, then it just changes where expression is happening, right? It's a really simple model to explain complex evolution through gene evolution and tandem. So the antennapedia complex and bithorax complex are far away, so that's what this thing means. Like they are physically separated in the genome, but within a complex, they're really close to each other. So some people just really are interested in studying this for the sake of it's a really interesting developmental model, but I think most people are interested in this because of Hox genes. So Hox genes are a cluster of developmental genes that are very, very similar to the clusters in Drosophila. How are they similar? They're not homologs. And antennapedia complex, like the gene, like sex chromosomes reduced, does not have a homologue in humans. But what's really similar is how they're arrayed in the genome. So in mammals, you have 4 clusters. They show this similar pattern of tandem duplication. They are similar in sequence, though I don't think they're homologous. So you can study Drosophila development just to study genetics, or you can study it to make inferences about human development. The genes will not be the same. The mechanisms will be very similar because of shared evolutionary history, shared kind of development from single-celled organisms to multicellular organisms. So that's the main point of kind of these slides. So the last point in kind of developmental genetics is what scale are we dealing with? So you've kind of seen it as we've gone through, right? First, you're setting up major poles in an embryo, and then you're setting up segments in a larva, and then you're identifying structures that will be in the adult animal. So there's really a cascade of gene regulation. So that's what this figure is. When a scientist says cascade, they mean like all these arrows pointing to each other. Right? It's like a waterfall. They're kind of pointing down, 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 down. And what does this do? It kind of defines successively smaller structures. So at the top of this cascade, you're giving it the biggest definitions. Front, back, top, bottom, right? 4. You're defining 4 regions of this embryo. And the next step, now you're doing the gap. head, thorax, abdomen. In the next step, with the pair rule genes, you're giving segments, individual identity, and polarity. And then finally, you're adding the expression of the homeotic genes to say, okay, you're in the thorax, you're going to be a leg. Or you're in the thorax, you're going to be a wing. Right? All of this is happening really, really early on in development. And it's all due to gene regulation, overlapping expression of a small number of genes. Does that make sense? So we're going to transition to another example, right? The whole point of developmental genetics is to get you to think about gene regulation in the context of molecular mechanisms. I know about genes, I know about gene regulation, I need to bring them together in a model to make predictions. So that's what we did a lot in class discussion yesterday with floral development and Arabidopsis, right? So question 2 was taking the ABC gene model that looks like that little pyramid and applying it to make predictions. What can you say about phenotype in this model? So this is Arabidopsis. It's kind of just a boring looking plant. But in your book, they describe kind of development in these concentric rings. And so I was talking about these structures. Maybe Dr. Drew showed you a picture, but we can just label them. So you have sepals, petals. That's an anther on a stamen. The carpel would actually be internal to the plant. The carpel would be kind of the whole thing, but definitely where the seeds are. But this kind of fuzzy looking thing, that's the stigma, which is part of the whole, people call it female reproductive part of the plant. So this would represent the carpel. Carpel's the whole thing, stigma style, ovule bulges. This is another model that's super similar to the homeotic genes, where the expression pattern is not kind of static, it's really like tension. That's how I like to describe it. So this is the wild type scenario on the left, and then some of the mutants that you encounter and how it changes the world pattern. And people doing experiments where they would evaluate mutants came up with this model, right? So this model is kind of like a hypothesis. Data support this hypothesis, but this is how they can represent kind of this complex three-dimensional structure with expression, this kind of pyramid. And so it's not that the lack of class A genes gives you stamens, it's the presence of class 3 or class C. You can think of it as like this gradient is being established this way, and this gradient is being established this way, and they kind of meet in the middle, such that if you inhibit class A in any world, class C is going to move, right? The morphogens, whatever you want to call it, are going to move and give the identity of that world whatever expression of class C will be. So we saw a lot of examples of that yesterday. So I'm just going to go back to this one really quick. So in a class A mutant, right, class A is usually expressed in world 1 and world 2, giving sepals and petals depending on the co-expression of class B genes. If you remove class A altogether, where the A would normally be expressed, now it's just all C. Right? So you could draw this one here, and like that pyramid shape would just become this. So we get carpel in 1 and 4. That's when C genes are expressed by themselves. And we get stamens in world 2 and 3. That's when B and C are together. Okay? We can take the wild type model and draw a new molecular model. That this is our hypothesis. To generate a flower that looks like that, we're saying that there is no class A, and we can draw this new model. So I'm not going to spend too much time on this since we did a long discussion. But just to kind of cement what we're thinking about this model, I'll open this question, then I'll go back to the picture. So you can rely on the picture. So what type of flower structure would you expect to see in a world 1 through 4, a mutant plant that failed to produce both in class A and B gene products? So here's your model. So the question was asking no. Okay, yeah, so majority of people are answering C, which is carpal, 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 carpal. Or
That's
D,
not
excuse
it.
me, carpal, carpal.
On
These
ours,
don't
it's
match,
different.
right?
Ours is different.
Yeah, they don't match, it's fine. The best answer is carpal, 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 carpal. So on the clicker, this would have been C. So it's kind of like a softball one, but it's supposed to demonstrate something. That it's not the absence of worlds. If you eliminate expression A and B, it's not that you only have two worlds, you still have four worlds, they just all have the same identity. It's very, very similar to homeotic genes in animals. You're not changing the number of segments, you're not changing the number of worlds, you're changing the identity by differing gene expression. Through just 3 genes, you can get lots of different structures in plants and floral development. Any questions on that? So the last part of development that we're going to talk about that will also help us transition into cancer genetics is cell death. Sometimes a structure is needed, or like a certain type of cell is needed in development, but then it's no longer needed. So you kind of build a structure, but then there's no longer space for it or purpose for it, so it has to be kind of decommissioned. That is happening through cell death. Not all cell death is regulated, but when it is regulated, the cell can repurpose, right? You put a lot of energy into a cell. If you want to kill that cell, you want to recoup some of that energy. So there's a lot of mechanisms to control cell death purposefully. So that's called apoptosis. So some people call it controlled or programmed cell death. Right, some animals might have the same digits we have, but might have extra tissue between them. That's cell death, right? We all in embryo, in utero, have tissue between our digits. Then there's programmed cell death that removes it. You have to build all of the tissue to allow bone and muscle to form. If you don't have tissue in between your digits, development doesn't occur in a specific way. It occurs in a different way. But if the tissue is gone in some animals, not in others, it's due to the differences in programmed cell death. So what is the opposite of programmed cell death? That's really necrosis. So this is often caused by injury. And it's often uncontrolled. And what we mean by that is it's not a cellular process that's signaling death, and sometimes the cell has a more difficult time of recouping kind of what's inside of that cell to repurpose it. Sometimes this could be purposeful, right? Like immunity, there's a lot of apoptosis that goes on with immunity, but again, this is kind of from the perspective of the organism itself. If you're killing a bacterial gene, or excuse me, a bacterial cell or a viral particle in your body, that's not apoptosis. It's more similar to necrosis, but that's kind of a separate set, because that's a foreign cell, right? That's not a cell that's endogenous to the animal's body. So we're not going to talk about all of this. We're going to talk a little bit about the regulation of apoptosis and a couple examples for development, and then a couple examples for disease, because then that translates to cancer that we'll do on people. So this is the figure in your book that's trying to compare and contrast programmed cell death and uncontrolled cell death by necrosis. So the big take-home point here is that they're distinct at several levels. So in apoptosis, you have the degradation of DNA. So it says cytoplasmic nucleus shrink and the nucleus fragments. You know, it's kind of, we can try to say there's purpose behind that. Again, purpose is always a hard part of biology. I think it's better to think about consequence. What might be a consequence of nucleus being in fragments? So we're right here at number 2. Yeah, that's what I was thinking of, the last part of what you said. So it's not like this is purposeful, the cell does this for this. I mean, it's probably over time selection made a system that has this consequence. So what Robin said, if you're fragmenting your nucleus, it prevents this cell that you're channeling or programming for cell death to be able to replicate, right? If it doesn't have all the DNA, it cannot continue replicating. So that's a really critical point of apoptosis. Fragmenting the nucleus to stop any potential of cell division. So we'll see this model, and then next week I don't know if we'll see this exact same slide, but we'll think about what can cancer do? Cancer is able to bypass this, right? Cancer is often super polyploid. Cancer cells have wildly varying numbers of chromosomes. They kind of can override this checkpoint of having to have an intact nucleus. So the consequence here is stopping cell division. There's many ways to stop cell division. I just wanted to point out this one here. Okay, then you have macrophages and vesicles engulfing things, and then phagocytosis. So this is the recycling part, right? Vesicles and macrophages can store and transport things across the site. So if you're not enveloping kind of the cell that's breaking apart in these macrophages and undergoing phagocytosis, you can't recycle what's in there. You can't recycle the nucleotides, the proteins, the amino acids. All of that's kind of lost to whatever environment that the cells are living in. So then in the necrosis, it's often, if it's injury, how do you injure a cell? Most of the time you're thinking about disrupting the cellular membrane, right? The cellular membrane is really important to keep a pH, internal, different than external environment. If you change pH, you get flow of solutions across osmotic gradients, right? So you see cells burst and lyse. So the injury here is often to the cell membrane. So it's not that there's something intrinsic to the cell to say swell, right? It's not controlled. It's not programmed. It's just a property of chemistry. If you mess up a pH gradient, water is going to flow differently in an osmotic gradient, right? And then there's no macrophages. You can't recycle. So in the previous slide, I was saying it's uncontrolled. You can't recoup what was in the cell. That's because the macrophages aren't around, right? The programming on the left-hand side of apoptosis is coordinated. Another way to think of it is coordinated, right? Macrophages are recruited to be able to do their job. In an injury, it's stochastic. Macrophages maybe are hanging around. Oftentimes, that's not the case. So the last important part about development is that, and I'm kind of trying to I've been trying to hint at this as we've been going through, but it really is about evolution at the end of the day. So we have common genes and developmental pathways. And how is evolution going to occur? If we're saying that the genes are the same and the pathway is the same, how is evolution going to occur? And mutations that are doing what? Yeah. What are the mutations doing? Yeah, so I was really thinking about change in gene expression. Our theme for epigenetics, development, and cancer is really thinking about gene expression, right? That's what's linking all of these three subjects. There might be mutations, but they're not changing how a protein functions. They're changing where it's expressed, when it's expressed. That's happening in development, but it's also happening on larger type scales that can give you evolutionary units. So we saw this yesterday in discussion. You also read an article about it. Mexican tetras. This is a story about kind of all of these things. It's a story about evolution. It's a story about genetics. It's a story about epigenetics and gene expression. So these animals live in a cave. They lack eyes, right? You can't really see it. They lack eyes. They actually start developing an eye, but what happens to it? It undergoes programmed cell death. The cells, the progenitor cells that would normally form, like in insects, we call them imaginal discs. I forget what you call them in vertebrates. And it's basically like a cluster of cells that will then give a structure. Those undergo apoptosis. Programmed cell death due to increased signaling of sonic hedgehogs. So it's not a change in the coding sequence that we saw yesterday in discussion, it's a change in the regulatory sequence, right? This paper demonstrated a link in expression levels of sonic hedgehog to eye loss. How is this a story about evolution? Cavefish, so there's two things. There's surface fish, cavefish, right? You guys know a little bit about phylogenies. If we have surface fish, I don't know where I'm going to draw this. Let's see. Surface fish 1, surface fish 2, cavefish 1, cavefish 2. What phylogeny do you want to draw? Or you don't want to draw any phylogeny ever again.
Right.
If you were just basing on what these fish look like, you might be tempted to do this, that the surface fish form a monophyletic group, cavefish form a monophyletic group, and then in some distance they had a common ancestor. But that's not what happens. When people actually do genomics of cavefish, they often find a pattern like this. These caves are being repeatedly colonized by fish from the surface, and then each cave independently undergoes changes in gene expression that causes the loss of eyes. To me, that's really fascinating, right? It gets at the common developmental mechanism. It gets at selection. Like if it's happening repeatedly, it's not just stochastic. There must be some type of selection which favors not having eyes in a cave. And you can understand the changes in gene expression. We said that there are changes in Sonic the Hedgehog. These cavefish do not share mutations in Sonic the Hedgehog. Each mutation is independently derived. So you learn a ton about Sonic the Hedgehog through all these different mutations that are accumulated. Any questions on that? Okay, I don't have many more slides. I'm just trying to stall so you don't leave too early. Okay, well, at least we have one person cheering me up. Okay, so let's just transition at the last part for immunity and genetic rearrangements. So this is kind of put in the developmental genetics chapter. I think it's a weird place there, but then when I started thinking about it more, it's all about gene expression. How are we going to change a protein, right? Like all of the different antibodies or kind of receptors in your cell are there. We've been talking a lot about changes in gene expression. What's the other way that we can change things in the genome? Recombination. So I'm kind of giving you the take home now. This whole section is about recombination. If you want to generate diversity when you only have so many things to work with, you mix them up. If you can't do it through gene expression, you do it through recombination. So for immunity, there's a lot of just different terms, so I'm just going to define these, so when you see the figures in the book, you know like kind of which part is which. So the first thing that we have is an antigen. So this is the molecule that elicits an immune reaction. So this is like the broadest definition, a molecule. Maybe it can be a protein, maybe it can be a DNA or an RNA. So we're just going to call it a molecule. Something is detected that's going to elicit an immune reaction. And then you have an antibody. So these are proteins. These have a more specific definition. So they're a protein that binds to the antigen and mark them for disruption. Macrophages, phagocytosis cells, or phagocytotic cells are involved. I do want to draw a distinction about programmed cell death because, again, this doesn't have to be a cell, it's just a molecule writ large. The other reason you might want to encapsulate something in a vesicle or macrophage is not so you can take the material, but so you can expel it, right? Vesicles can go to the cell membrane and then expel stuff. So how is this all organized? I'm running out of room a little bit. So we have kind of the humoral immunity. So this is production of antibodies by B cells. So some of you might know some of these terms probably better than I do. And then you have cellular immunity. These are T cells. And then in kind of classifying these immune responses further, you can kind of think about clonal selection, right? So you have a cell that makes antibodies to an initial infection, and then sometimes those cells undergo clonal expansion and then are incorporated to give you cellular memory or like immune memory. So that upon subsequent infection, you don't have to make an antibody de novo, you kind of already have the memory. So that has a really interesting parallel to cancer because cancer is all about clonal propagation of cells. So this is what it looks like. So the reason for putting all those definitions up is so that when you see a figure like this, you can start to piece it together. So B cells are part of humoral immunity, right? They're made in marrow. T cells are made in different cells, cellular immunity. This is the example of the thymus. Both of them have to be able to interact with the antigen in some way, or interact with the molecule that has the antigen attached to it, because it might be a whole cell in the case of a bacteria. So then you have antigens, right, or the molecules that signal that this is foreign usually, and then antibodies are being produced. So what do you notice about this picture? On the right-hand side, so ignore where the cells are coming from. That's really important for like how long immunity lasts and where it's coming from. But I want to focus on the right-hand side. So what is something you notice about the right-hand side? Or you might already think about if you've thought about immunity. Yeah, and how is that interaction facilitated? Sure, but yeah. But what do antibodies in this picture? They look a specific, something about them, right? Shape. That's when I see a picture like this. What the authors of the textbook are trying to get you to think about a shape, matching, right? So everything you guys said is 100% true, but if you take a step back and just think about a general principle, it's all about shape and matching, right? That's how certain diseases get around your immune system. They change something, the antigen changes, and then you have to develop new antibodies. So this is the selection about, this is the slide about clonal selection. So I'm not going to go through it super in depth. But basically, if you're making a bunch of cells, you can rapidly produce similar cells through what's called clonal propagation or clonal selection. Selection is kind of like a misnomer here. But what it means is that if you make a bunch of cells, some of them are going to go into the cell, right, to destroy. But if those are successful, your body has a way of storing them, right? So these cells here, they're all clones. They're genetically identical. Most daughter cells are not genetically identical because of crossing over. Well, that's only in the gametes, right? Well, not really. We know mutations occur not through crossing over, but just through DNA replication. This is clonal, right? This has much different rules and thresholds for mutation. So what that gives you is that in the secondary immune response, you're kind of just making another batch of clones. So in cancer, so this process that's happening in your body normally is very mutation kind of intolerant, I guess. Cancer is the opposite. There's a lot of mutations happening in clones that generate clonal disease. So this is the shape part. So here's an antigen binding site shown here, and then it's three-dimensional structure. We show them with those little kind of silly stick cartoons, but they have this three-dimensional structure. They're mostly characterized by that Y-shaped structure. So this is immunoglobulin. So there's 4 peptides, a polypeptide chain. Okay, so you don't need to memorize the structure of immunoglobulin. You don't need to know that it has 4 chains, but you need to know that it has shape. Shape's important. How are we going to make new shapes? So this is my last slide. We're going to make new shapes by somatic recombination. Right? We saw recombination and crossing over in terms of kind of gametogenesis, making haploid gametes that will come together to fertilize. That is not occurring, like I just said, in somatic cells or cells that are not part of the germline. So how are you going to recombine antibodies to make, you can wait for a mutation, that's a really slow process. The quickest way to make new antibodies is through somatic recombination. So that's what it's just showing here. So I'm not going to walk through all of this, but basically, right, in eukaryotes, we have introns. If you have duplications are a great way to recombine because they're homologous sequences that vary in length, so that you can make more copies that can undergo mutation and kind of mix them. So just in case you're interested, this is gene segments. For human haplolysis. Right, immunoglobulins have 4 chains, 2 heavy, 2 light. This is happening at most of the chains, but more specifically in the light chains because they have less functional structural constraint. Right, that heavy chain has the Y part coming down. You can't really mutagenize that because then you lose the structural integrity of the immunoglobulin. Those little light chains can be recombined much more efficiently. Okay. Any questions on this? So I'm going to give you 15 minutes on the quiz. If you want to stay here until 9:45, you can. We're done really, really early. So when you're done,